Hello everyone. Today if I remember, eighth or maybe even ninth lesson, I'll need to check it out. So, and today our guest, Professor Irina Makarevich. She's a professor biology in Hamline University in St. Paul. So I'm also from Minnesota, but because of this coronavirus problem, so each of us sit at home. And Irina today will give us a lesson about proteins, about their design and structure. And as illustration, she will show solving puzzle in a program Foldit. Hello, Irina.
Hi, Anatoly. Hello, welcome to this lesson, and I hope that you will like what I'm doing. So I'm going to share the screen and first tell you a little bit about the protein structure and how proteins are folded. And I hope that you can see it now. So proteins are really cool molecules. They conduct all the chemical reactions in your bodies, essentially. They build your cells in many different ways. And it's amazing to think that these proteins are built only from 20 amino acids, from 20 smaller molecules. Those 20 amino acids are all very different in their structure, and their order is really determining the structure of the protein and the function of the protein. So the order of those amino acids is determined by your DNA. And DNA structure defines how those amino acids will be connected to each other. The order of those amino acids, we call it primary structure. And essentially it's just amino acid 1, amino acid 2, amino acid 3, and so on and so forth. Proteins vary widely in terms of how many amino acids could be parts of those proteins, from very small ones like 20 to 50 to really, really large ones with over 1,000 or even several thousand of amino acids. Biologists usually say that primary structure determines the structure of the protein and its function. And so from that perspective, DNA sequences determine the structure and the function of those proteins. The problem though now is, how do proteins do that? Imagine this really, really long molecule with thousands of amino acids put up in order, and they have to fold correctly in these three-dimensional structures. For example, to form polypeptides or smaller proteins, and then even such big proteins as hemoglobin that is shown over here. It contains 4 different protein molecules arranged together. And again, remember, all of this is determined by its primary structure. How does it work? It's actually a very interesting question, and we don't know exactly all of the details, but the ones that those amino acid molecules form with each other, that's what determines how the proteins will be folded. It all starts locally with several amino acids that are located close to each other, forming some sort of a structure. Those structures typically are either alpha helices or beta sheets. And you can see that they look very different, but the molecular forces that actually bind them, they're similar. They're called hydrogen bonds. So in those amino acids, look here, there is a hydrogen atom, for example, on one of the amino groups of one of the amino acids. But then there is an oxygen here in another amino acid. Well, this oxygen carries an extra electron pair, and hydrogen atom really wants to interact with that electron pair. When I say wants to interact, we are giving those molecules human properties, and it's really fun to think about it this way. But in reality, when these guys interact, the energy of the molecule goes down and the molecule becomes more stable. So in many ways, it's really an energy game. And the other properties of the amino acids, for example, how many of them are hydrophobic or hydrophilic, determines whether the molecules, those amino acids, will be forming alpha helices or beta sheets. If the words hydrophobic, I'm sorry about that, and hydrophilic are really confusing right now, don't worry about it. But if you want to learn more about them, you can either Google and look it up, or we will try to talk about it in another video, and I hope that you will join us for that discussion later on. So when the molecules form those alpha helices and beta sheets, then the whole molecule needs to be folded correctly so that all of those local structures are organized relative to each other correctly. And that energy game goes on. This funnel is how many scientists think about those energy games and those folding games. as you can see here, as the molecule falls into that funnel, its energy goes down. And again, remember, less energy means more stable protein. In many cases, there are many different stable structures, many different ends to this funnel. These funnels usually don't have just one end, one bottom. In some proteins, like as shown in this example, only one of those stable structures is actually a good stable structure. It produces the protein that is functional. And the other two in this case, or maybe even three if you count this one, produce proteins that are not helpful for the cell. In this case, those proteins form some aggregates and even amyloid fibrils. This is an example of the proteins that can cause Alzheimer's when folded incorrectly. But in many other cases, multiple bottoms of this funnel, all of them are good and functional, and the protein can switch from one structure to another depending on the situation in the cell. So again, when the protein gets folded, its energy going down, and the more energy favorable, less energy situation is usually the one where the protein is most stable. And this is a good point to switch to the program that is called Foldit to show how that works and just to actually see a good example. Let me switch. Oh, question.
Just quick question. So since there is a few minima, are how the proteins knows which one is it's functional in the sense of working in an environment as it should be? What helps to protein to select the right minimum?
This is a really good question. And for some proteins, we don't really know how they're functioning, and they frequently go from one situation to another. Maybe the protein can be bound to another molecule, for example, to a short phosphate addition, right? Or it could be another type of additional molecules that helps protein switch from one position to another. Sometimes proteins bind to other larger proteins, and that binding helps the protein switch from one position to another. There are proteins that are called chaperones. They are very special proteins. They're protein helpers, and they're really cool. They help usually bigger proteins to fold correctly. Those chaperones are also called heat shock proteins. So when the cell gets heated up, when the temperature goes up, many protein folds break. Just think about what happens when you boil an egg and how egg white changes, right? That's a protein that becomes really, really screwed up. But when the temperature goes up just a little bit, those chaperones, so heat shock proteins, help proteins find the right shape. They create the environment where protein can be folded correctly. Did that answer?
So basically, if I understand, so they worked in the teams.
Yeah, exactly. Yeah, many proteins work as teams. When you think about protein folding too, when the protein is just being built, there is no the whole protein yet. So it's another really interesting thing to think about, how the one part of the protein needs to be folded correctly while it still doesn't know how the second part of the protein is going to look like. It's folding in the process of being synthesized, and chaperones help there as well.
All right, thank you so much. Sorry for interruption.
Oh, no worries, that's fun. Let me go back to Foldit. So Foldit is a cool program that you can register for online for free and become a part of this community of people who play games with protein folding. And if you start, you can start with their campaign. It's essentially just their several puzzles, well, 39 nowadays. You can see that I only completed 38 of them so far, that kind of start you on this adventure. And we will work today on one of those campaign puzzles. After you're done with them, you can start working on science puzzles and help scientists solve various protein folding problems. In fact, they had several really good successes with HIV protein folding and several others that you can read on the Foldit website if you are interested in seeing it more. So we will be working on this sheets and letters campaign. Come on. No, I can't start it. Feeling stuck. No, I'm so, oh, see, like, I don't know what's going on now. They're gonna have to redo this.
Yeah,
I have no idea. Like, it's like Foldit does this once in a while. And I don't know, like before that, I clicked on it 3 times. I'm just clicking and it doesn't start it.
Okay, maybe just
But see,
restart.
like, okay, so now it started a wrong one. Okay, we'll redo it. But was it okay overall, what I was talking about?
Yeah, just go restart it. So it's yeah, it's happened to me several times. Or and also there are people who use it shouldn't be frustrated by once in a while. I think one of their you do it online, right? Version.
Yeah.
Yeah, that's why that's one of the reason might be because if there is a lot of people working on that, so that might create they just restart it again and try to start this puzzle. It happened to me also several times when I needed to restart Foldit. One of the solution I found it, and when it was with a puzzle. So since you're not related to the community, even they not recommend it, I would usually use the local version. So without going to, not internet, not non-internet version. Maybe that would,
Yeah.
let's go and see if it's, just try to start their not internet version. So locally on your computer, not a big deal.
Okay, I think it's now hopefully working. All right. Let's see if I can make it work now. I have to share screen again. And here we go. Yay! Sorry about that, but at least we are back
Well,
to where
actually,
we wanted to
that's a
be.
good example that it, it may happen when the people work with a program.
Yeah, unfortunately it happens. And again, I'm sorry about that. But this is an example that is called sheets and letters. As you can see, there are beta sheets in here. Here is one, here is another one, and here is the third one. And so now the job here is to try to get all of those protein parts together. Foldit has several tools that are kind of sort of automatic, and I wanted to show you one right away. So I'm going to say wiggle. What essentially it means is that the program is trying to find the best optimal structure for this protein. And you can see that it forms some of those hydrogen bonds. Do you see that? Like there is another one here. So it forms some of them, but it will stop improving. So look at the score. The score is positive, but remember it's all energy gain. So these numbers are really negative. We are trying to dig down, not go up the mountain. And as you can see, the numbers stopped changing. And to get to a good situation, to get to a stable protein, we wanted to get to a lot lower number over there. So what's going on here is that this protein found more or less stable bottom in that funnel. But that more or less stable bottom in the funnel is not really the one where we want to be. It's not one of those more like really true stable situations. And the folded protein program by itself cannot just solve this problem. So what we need to do is we need to tell it, hey, we really want those guys to be together, those beta sheets. And it's okay that you don't like them, but we really want them to be together. We can do it in folded program by building those rubber bands. I'm just clicking on a shift and kind of pulling things together. What those rubber bands say to folded program that, hey, those beta sheets really need to be together. And then I'm going to try to wiggle again. And as you can see, they are not separated anymore. And some of the other hydrogen bonds are forming. And these guys are really, really close to each other now. I can even rotate it, see, and they're closer, but the number is still really small. So what happens in here, I will stop the wiggle, is the initial place where just wiggle without rubber band got this program to just wasn't very good. And it created barriers so that now this puzzle cannot be solved from that position. We need to start from the beginning. Well, it's not a problem because there is a reset puzzle bottom. So we can just go there. We can say reset the puzzle. And now I am going to set up those rubber bands right away. I'm just going to say, I'm serious about those beta sheets. They need to be together. They need to be forming those hydrogen bonds. And even if they will be too close to each other, it's still okay. So I'm doing that. you might experiment for setting up more of them or smaller number of those rubber bands. The protein probably will be working in either case, but I set up 4 of them in both of the cases. Let's try to do wiggle. See what's going on? Now my hydrogen bonds are really happy to be formed. My score now becomes really, really high. Again, remember, it's actually really, really low. And I solved the puzzle. So I'm just going to say stop.
All right, congratulations.
I am going to stop sharing. And
Uh,
I'm sorry about that debuggle, but I hope that you enjoyed playing the game.
actually less than a minute, and I think it's kind of useful so that people know something may happen on the road, like with your puzzle solving. So you went to the wrong way, getting the local minimum from which the program couldn't take it to the right minimum. So you needed to go back, and that's what happens sometimes in our life. So we think one way is good, so but we come to the wall, say, okay, we need to go back. Thank you so much for sharing your knowledge, Irina. Thank you very much for their lessons. So
Thank you for including me.
actually, it's a good idea. Maybe I will discuss with you and maybe discuss separately hydrophobity and hydrophility. I think it's very interesting concept for such a lessons. Maybe we'll do it in the future. Thank you so much. Goodbye.
Thank you. Take care.
See you again.